the activator proteins and the repressor proteins, including the ones that change euchromatin to heterochromatin and the DNA methylation, all of those affect whether the mRNA is going to be made or not, or I should say all those affect whether the primary mRNA transcript is going to be made or not, gene regulation at the transcriptional level. But that's not the full extent of gene expression. For complete gene expression, that primary transcript has to be spliced, the introns have to be removed, you get the poly A tail and the 5' cap to get the mature mRNA, then it has to exit the nucleus, and out here in the cytoplasm, you might remember there are enzymes that can degrade mRNAs, so it has to survive those enzymes, then it has to meet the ribosome, and the ribosome has to scan along it and completely scan along it from start to finish to get the start codon and the stop codon to assemble the protein. So that's the translation step. And so at each step in this procedure, there's an opportunity for the cell to regulate transcription. And since each of these events, like the splicing and the RNA being degraded, the rate that the RNA gets degraded, and the ribosome being able to bind onto the messenger RNA and scan along it and make the protein, since each one of those steps is controlled by enzymes, by changing the level of those enzymes that carry out that step, or by modifying those enzymes, the cell can regulate the expression, how much of the actual functional protein gets made. And yes, most of the expression, differential gene expression does take place here at this transcriptional level, but for each of the steps, they found instances where the cell is modifying that other step, some other step to regulate the expression. As I wanted to go through a couple of these other steps. Remember about the introns being removed. That takes place here in the nucleus. And let's see, that in the gene, you find exon regions and things that will be transcribed into intron regions. And so the primary transcript has those. And then these SNRPs, these proteins and nucleic acids found in the nucleus, bind onto the exon-intron boundaries, and they get joined together, and the intron gets looped out, and the two exons get spliced together, and that's how you get the mature mRNA. Like you notice the two red blocks there, the exons get spliced together because this intron is removed, like in that zone you see right there. But remember, many RNA transcripts, primary transcripts, can undergo something called alternative splicing, where the actual pattern of which introns are removed can vary from one to another. For instance, this exon right here can sometimes be joined to the green exon right there. Sorry, my laser looks like it's dying. Or sometimes that exon there can be joined to this purple exon right there. And that's alternative splicing, and it gives you two very different proteins because you get very different exons being used. Well, so how do the SNRPs and the spliceosomes decide which pattern of intron removal to do? Well, it's not fully understood, but there must be some enzymes, there must be some proteins that direct the SNRPs to either make this transcript or to make that transcript from the primary transcript. And so by varying the amounts of those controlling enzymes for the SNRPs and the spliceosomes, that is a type of gene regulation. It does affect how much of a certain protein the cell is expressing. Oh yeah, so there's in purple you see the splicing pattern. That or that can be regulated, and so that is a type of differential gene expression, and they have found instances of cells varying their alternative splicing depending on their conditions. Once the mature messenger RNA makes it out into the cytoplasm, the ribosome has to bind onto it and then scan along it all the way from start codon to stop codon before a functional protein can be made. And if anything blocks the ribosome from scanning along the messenger RNA, that's going to block expression of the protein. And so there are some proteins that are found in the cytoplasm that can do exactly that. They can block the ribosome from completing its translation step. So there's an mRNA, and imagine this thing right here is one of these blocking proteins. It can bind onto messenger RNAs, and so when the ribosome comes along, you're just not going to get the complete protein out of it. It's going to stall the ribosome. That's yet another level of differential gene expression by blocking the ribosome. And there are also enzymes in the cytoplasm that degrade mRNAs. mRNAs have a finite lifespan in the cytoplasm because these enzymes can destroy them. That, in a sense, is another way of regulating gene expression. Imagine this now is an RNA degrading enzyme. By attaching to the messenger RNAs, shortening their lifespan, it chops them up into bits, and that will also affect gene expression. Now, if you think about what I just told you here for the proteins that block RNA transcription, translation by binding to the mRNA, or the proteins that degrade the mRNAs, again by binding to the mRNA, I didn't say anything about specifying certain RNAs and leaving other RNAs alone. I just presented these, that the cell is going to block all the mRNAs, or the cell is going to degrade all the mRNAs, but I didn't talk about selectively doing to some mRNAs to another. But that's really the essence of differential gene expression, how certain genes, the expression of certain genes can be turned on or turned off. And so what we might be interested in asking is there a way for the cell to block only certain mRNAs but leave other mRNAs alone? Or is there a way for the cell to degrade certain mRNAs but leave other mRNAs alone? That would be the essence of differential gene expression. And the fact is that they do find that. It's called something called RNA interference. But the end result is blocking or degradation of just certain mRNAs, but not other ones. And to illustrate this, let's use this as the mRNA that the cell wants to target for degradation or blocking. But let's imagine there are other mRNAs floating around also that the cell doesn't want to change the expression of. Well, we know the difference between mRNAs coming from different genes is they have different sequences, which makes sense. If the mRNAs come from different genes, we expect them to have different sequences. And so I'm going to say that this mRNA that the cell wants to downregulate has this certain sequence, GU, CU, and other mRNAs don't have that particular sequence. And so what the cell is going to do is it's going to target the specific sequences in the mRNAs that it wants to block expression of or downregulate expression of. And I'm running a little short of time. Well, I'll pick up on this on Wednesday, but it's another area that's just newly emerging in biology, how the cell can interfere with certain mRNAs and not others. Okay, well, let's see. So put your goodies away, and we'll do the quiz.